Good morning, everyone. Hope that you and your families are all doing well during these very difficult times. First, thank you and congratulations to Thomas and Yabing and all of the members of the program committee. The program is incredible despite all of the challenges that COVID-19 has brought to us. I'm so very pleased that we're able to spend some time together and share our ideas with each other virtually. I want to thank you for the opportunity to talk with you today. I hope that I'm able to provide some constructive ideas that put us on a good path for advancing science and improving the lives of people around the world. I also hope that I can make you smile a couple of times along the way. With that, I will begin my talk. I will talk to you today about our most important discovery, the research question. We often debate how to design or analyze a study, but at its core, what we are really debating is not what the answer is, but what the question is. And once the subtleties of the question are well defined and understood, the path to the answer becomes clear. Unfortunately, we fail to recognize that many of our traditional approaches to the design and analysis of trials do not address the most relevant questions for clinical practice. And identification of these most important clinical research questions and adjusting our approaches to address those questions is an area where we can greatly advance medicine and public health. So about 15 years ago, I started teaching a clinical trials course at Harvard, and I gave all the students a questionnaire on day one. And these were primarily early career MDs who were starting clinical trial careers. And at the end of that questionnaire, I asked them to complete this sentence: a statistician is blank. And I thought I'd share some of the answers that I received over the years, some insightful and some humorous. So a statistician is to data what a doctor is to a patient, somebody who's wrong 5% of the time, a coupon that gets me 15% off my sample size, my best friend when writing a grant proposal, someone who answers my questions with more questions, the gateway to understanding, a scientist who transforms data into knowledge, an angel of God, an unlikely bedfellow that destroys dreams, the rate-limiting step in manuscript writing, and the wizard behind the curtain. Perhaps elements of truth in all of those. So I'd like to start off with a quick SWOT analysis of clinical trials. Certainly the strengths of clinical trials right at the top is that we have these phenomenal tools, the best tools in our toolbox, randomization, which is the foundation for statistical inference, blinding, control groups, prospective observation, the intention to treat principle, which protects the benefits of randomization and assesses pragmatic questions. On the flip side, the weaknesses are, one, they're quite expensive, time-consuming, and resource-intensive. Opportunities, and this is where I would like to focus most of my discussion today, is I think we have a great opportunity for pursuing greater pragmatism. And by here, I mean pragmatism, finding relevant answers for clinical practice. That may or may not include using real-world data. So here I'm not necessarily referring to use of real-world data, although it could be. So I would like to spend most of the time talking about this particular opportunity. However, I'd like to make a few comments about threats that I see happening, and two in particular. The first threat is we all have an innate desire to do things faster and cheaper. And this desire is often magnified by business and political pressures. And although this is understandable, these desires can be dangerous and threaten objectivity and result in studies that have lower applicability, lower integrity generally. A second threat that I'm worried about is we seem to be quite susceptible to commercialism for trendy methods. and the methods are often labeled as innovations, but these innovations often actually lower the evidentiary standard and introduce greater uncertainty rather than getting us more robust answers. So these innovations, many of them, I think we need to be careful, ask ourselves the question about whether these are really progress or regress. 25 years ago, innovation was developing methods requiring fewer assumptions to improve robustness. The goal was to be model-free when estimating a treatment effect, because model-based measures are difficult to interpret and associated with assumptions that we may not be able to validate. However, today, many of the innovations appear to be compromises. They're basically a willingness to make more assumptions, perhaps to reduce a sample size. And this can be at the expense of robustness, objectivity, and scientific rigor. Now, it's true that our approaches are in need of improvement. This is a quote from Dave Demetz and Rob Califf, the former FDA commissioner. Most clinical trials fail to provide the evidence needed to inform medical decision-making. The implications of this are largely absent from discussion. And so can we redirect our motivation to find better answers to the most important questions for patients and clinicians that would better inform medical decision-making? And I believe we can, and I'll talk to you about some of those ideas today. And sometimes if we pursue these ideas, we're also going to get efficiencies in doing so. So I'd like to begin by telling a story. A few years ago, I got a leak in my roof, and it created a water bubble in my wall. And in addition to having to get a new roof, I had to repaper the wall. And I asked my neighbor, who had recently papered a similar sized room in his house, how much paper did you buy? And he replied, 6 rolls. Upon finishing papering the wall, however, I realized I only used 4 rolls. And I told my neighbor, I said, I have 2 rolls left. And he replied, oh, that happened to you too? And of course, the issue, despite my frustration, in some ways I had to absorb some of the blame for this, in the sense that I asked the wrong question. I asked them how much paper did he buy, not how much he used. And I think in many cases we need to pay better attention to the questions we're asking and answering. Perhaps we motivate this by a quote from Jonas Salk, who developed the polio vaccine many years ago. What people think of as the moment of discovery is really discovery of the question. And I think there's an opportunity there for us to do the same. So let me ask a series of questions that might help focus what I mean by finding better questions than the ones we seem to be addressing these days. In clinical trials, we define analysis populations. We often, for efficacy analysis, we use the intention to treat population. Safety analysis, we use a safety population. Those two populations are not the same. Then at the end, we may combine these analyses into some sort of benefit-risk analyses. But to whom does this benefit-risk analysis apply? We're actually estimating parameters from different populations and then combining them. If you think about what is the estimand, in some ways it doesn't even make sense. Question 2. There's been great work on estimands. We finally get people to realize and recognize that different analysis populations address different questions. But I'm not sure we're always choosing the right estimand and the right analysis population. So consider the following example. Suppose a randomized trial is conducted and we're comparing treatment A versus treatment B. A trial participant is assigned to A, discontinues A, and begins a new intervention C. The participant then experiences a serious adverse event. It's adjudicated as related to C, but not A. This leads to the belief that safety is not an issue for A because it's C's fault. Now suppose 10 additional trial participants discontinue A, begin C, and experience the SAE. Again, adjudication links the relationship to C, but not A. There's no such events in Arm B. Would you volunteer to be randomized into this trial? If you were on a DMC, can you allow continued randomization into Arm A? Adjudication is not really the relevant question here. The relevant question for the patients in this trial and for future patients is the strategy of application. And this is addressed with ITT types of analyses. We talk about this problem in a recent publication in Annals. Question 3. Suppose we measure duration of hospitalization. Shorter duration is better. Or is it? The faster the patient dies, the shorter the duration. So the interpretation of one outcome needs the context of other clinical outcomes for the same patient. If you give me a summary statistic of duration of hospitalization, I don't even know how to interpret it. Only when we understand the survival status of the patient does the number make any sense. So why do we analyze these things separately? Suppose you increase of risk my risk of death from 1 in 10 to 2 in 10. That's a relative risk of 2. That's very important. Now suppose you increase my risk of death from 1 in 100,000 to 2 in 100,000. That's also a relative risk of 2, but nearly irrelevant. Is relative risk what we really want? To further explore this relative risk issue, consider a clinical trial called the TALIS trials, published this year in New England Journal of Medicine. And it was a randomized, double-blinded, placebo-controlled trial, and it compared ticagrelor and aspirin versus aspirin alone in patients with acute ischemic stroke or TIA. The primary outcome was time to stroke or death at 30 days, had a hazard ratio of 0.83, significant p-value of 0.01. The primary safety outcome was time to severe bleeding, and this had a hazard ratio of 4, also a p-value less than 0.01. Is this too much bleeding? Sounds pretty high, this hazard ratio of 4. And if we start to think about, well, how do we combine these hazard ratios or measures of relative risk when assessing benefit risk? Well, consider the primary outcome of time to stroke or death at 30 days. There were 303 events in ticagrelor and 362 in control. So there's a prevention of 59 events. The safety outcome, there were 28 events, severe bleeding events in ticagrelor, 7 bleeding events in placebo. So it costs you 21 events. So there's a total savings of 38 events if you considered them somewhat comparable. Not saying that they are, but just as a quick aggregate analysis. But instead of the 28 versus 7 bleeding events, suppose that for the primary safety outcome, there were 10 events in ticagrelor and 1 event in placebo. Well, that's a hazard ratio of 10. And that sounds really bad. But the cost is only 9 events. And the total savings would be 50 events. And so combining these measures of relative risk is extremely difficult. And we have to think carefully about whether that's really what we want in primary analyses. To further relay the relative risk issue, suppose we conduct a trial and we have 500 patients in each arm, treatment A versus treatment B, and we observe 1 event in arm A and 3 events in arm B. So what is the confidence interval for the relative risk? Well, it's 0.3 to 158, very wide confidence interval. What about the risk difference? Well, the risk difference confidence interval goes from minus 0.4 to 1.2%. But instead of this trial having 1,000 patients, suppose this trial had 10,000 patients, and we see 1 event in arm A out of 5,000 and 3 events in arm B out of 5,000. What happens to the confidence interval for relative risk? Well, nothing. The precision by which we measure relative risk depends on numbers of events, and we didn't get any more events, so we say there's no additional information. But on the other hand, we just observe an additional 9,000 patients. How could this be no more information? But then when we look at, say, risk difference, we see a confidence interval getting much narrower, minus 0.04% versus 0.12 on the risk difference scale. Question 5. Suppose a loved one is diagnosed with a serious disease. You get to select treatment, and there's 3 treatment options. We'll call them A, B, and C. Now, for simplicity, let's assume there's 2 outcomes, and they're equally important. A treatment success, yes or no. Safety event, yes or no, both binary outcomes. Now, luckily enough, there was a randomized trial that compared these 3 outcomes, and so we'll analyze these outcomes. There's 100 patients in each of the 3 treatment arms. There's a 50% success rate in all 3 arms, A, B, and C, each have 50%. The safety event rate is 30% in A, 50% in B, and 50% in C. So which treatment do you choose? Well, they all have the same success rate. A's got the lowest safety event rate. B and C are indistinguishable. You can't tell the difference. So clearly we would choose A. But instead of using the patients in the trial to analyze the outcomes, as we just did in that example, let's flip that upside down and use the outcomes in the trial to analyze what happens to the patients. And if you think about what the patient outcomes are, there's 4 patient outcomes in this study. Either they experience the treatment success, yes or no, with or without the safety event, yes or no. So let's categorize or cross-classify these 4 different patient outcomes. Now, it turns out that in our trial, the safety event and the treatment success were uncorrelated. So there 35 of the patients experienced the treatment success without the safety event. But in treatment B, they were positively correlated so that no patients had treatment success without the safety event. Treatment C, they were negatively correlated. So there's actually 50 patients that experienced the treatment success and avoided the safety event. Now, let's move on to sort of subgroup type questions. A negative trial may not mean the treatment is no good for anyone. A positive trial would not necessarily mean that it works for everyone. So how do we identify the subgroup of patients for which we want to treat? Well, let's return to the trial that we just looked at with 3 treatment arms, A, B, and C. We could look for predictive markers of efficacy, and if they were very good predictive markers, what we would find are these 50 patients in treatment A, these 50 patients here in treatment B, and these 50 patients in treatment C. Noticing that only in treatment C do we actually find the patients that we should be treating. We could look at predictive markers for safety, and that would identify these patients. But of course, neither of these approaches by themselves identifies the patients necessarily that we want to treat. And then if we're going to head towards personalized medicine, shouldn't that be based on analysis of the patient rather than an analysis of a specific outcome? So the theme here is that the clinical trial arithmetic has never been quite right. Compare and combine is not equal to combine and compare. And it's comparing and combining is what we do. We take outcome number 1, we compare the treatment with respect to that outcome, we repeat that for all the other outcomes, and then combine. However, that's not the way patients experience these outcomes. What we need to do is combine outcomes within patient to get a global assessment of the patient, and then compare how treatments compare the treatments with respect to that global outcome. That's what's most relevant for evaluating the totality of effects as they occur on patients. As my father told me many years ago, the order of operations is important, and we've never had the clinical trial arithmetic quite right. Question 7. The effects of interventions are multidimensional, and they result in cumulative effects on individual patients. Why aren't we evaluating these cumulative effects? We're often evaluating, again, one outcome at a time, not how they might be piling up within patient. There was an NBA basketball coach named Frank Layden, had a player that was not producing. He just couldn't get the player to do what he wanted them to do. So he approached the player and he asked them, "Son, what is it with you? Is it ignorance or is it apathy?" And the player looked at Coach Layden and said, "Coach, I don't know and I don't care." And I feel like that although our arithmetic has never been quite right, I don't know if it's because people don't understand that the arithmetic is not right, or whether they don't care that the arithmetic is not quite right. But I think that if they don't know, we need to better educate them, and if they don't care, then we need to motivate them, because we're not getting at the question that is most important uh, for clinicians and patients. So we had developed some ideas for addressing this problem. Wrote a paper with Dean Folman where we say, well, let's use the outcomes to analyze the patients rather than the patients to analyze the outcomes. And it's a fancy play on words, but that play has meaning, and that this is a step towards pragmatism in benefit risk evaluation. And one methodology we've been using to approach this is called DOOR. And DOOR is an acronym for Desirability of Outcome Ranking. And the idea here is that before we analyze several hundred or several thousand patients, we must understand how to analyze one. How do we use the outcomes to analyze the patients? And to think about, there's a patient journey through the trial, and we analyze that patient journey through the trial, synthesizing the benefits and the harms that they experience, and the quality of life that they have along the way. So here's an example. This was an example that compared two antibiotic drugs, colistin versus an old drug to treat bacterial infections, colistin, versus Cazavi, which is a relatively new antibiotic to treat infections due to CRE, one of these superbug infections. And we created what we call a DOOR. The DOOR is the desirability of outcome ranking, and it's basically an ordinal outcome. And in this particular example, we chose a 4-level ordinal outcome. And playing on the desirability concept, most desirable in the top category there. The patient lives and they're discharged home from the hospital. The least desirable is on the bottom, where the patient dies. But there's a couple of layers in between where some things go right, the patient lives, but not everything goes right. They're not discharged home yet, or maybe they have some sort of a renal failure, toxicity. And so we have these layers. And the idea is that if colistin here is the old drug, it's the control group in the context of this analysis, that patients would fall wherever they fall into these categories. And that a new drug, what we're hoping for is that patients sort of migrate northward to more desirable categories. And then we calculate something called the DOOR probability, which is the probability of a more desirable result, global result, with respect to a global outcome when assigned to the new therapy versus a control. Now, this is quite a bit of a different metric for us in clinical trials. It's not a difference in means or proportions or a hazard ratio. However, once we can get some familiarity with this particular metric, it's actually very intuitive that if you have two treatment choices, if you're a clinician making a treatment choice or a patient who's got to make a treatment choice, choosing between A and B, wouldn't you want to know the probability of a more desirable global outcome in one treatment relative to another? That's a very practical calculation, practical metric, and informative metric for helping to guide clinical decision making. So this is what happened in this particular study. These were the way the numbers fell out in the DOOR categories for colistin and Cazavi, and observational studies. So we did some inverse probability of treatment weighting. The probability of a more desirable result with Cazavi was 64%, and you can get a confidence interval for that. Now, another type of analysis that could be done to that DOOR is to what we called partial credit, and where you could imagine an analog to an academic test. Whereas if you make it in the most desirable category, you live and you're discharged home, you get a perfect score of 100. If you die, you get a 0. But if you're in the middle categories, you're going to get partial credit. And so we've been working on these ideas. And this idea would allow us to account for unequal steps between the categories. And also, if we wanted to do some patient preferences, we could get patients could actually tell you how they value being in these different categories, and we can use things like quality of life information to help guide uh, um, partial credit scoring. So let me show you, this is not just an idea that we apply in infectious diseases. Here's an example from a cardiovascular event prevention trial. They often evaluate the time to the first event of death, MI, or stroke. But of course, there can be multiple events. It's not just a first event issue. And if we evaluate time to this first event, particularly if it's a composite, it would fail to distinguish the differential importance of these events. Death is more important than a non-fatal event. Something that is disabling is more important than something that is non-disabling. If it has permanent consequences, it's more important than something that is transient. So in deciding how to treat patients, shouldn't we be considering this information? So what we might do in cardiovascular event prevention trials, we see these typical endpoints: survival, stroke, MI, major bleeds is the toxicity. Might we set up a door that looks something like this? Most desirable at the top, the patient survives and has none of these events. The least desirable is the patient dies. There's a couple of layers in between. So the second category might be where they survive, but they have one event that's non-disabling. If they have more than one event, then they slide down again to desirability level number 3. If those events are disabling, then maybe they slide down even further. So and we might even consider this as progressive disease states, and we could evaluate moving into these categories somewhat longitudinally. And we talked about this in a recent paper published in Clinical Trials. Here's another example from antibacterial indications. Trials typically use binary endpoints. We have things like cure, which means that the patient survives, the symptoms resolve, this microbiological eradication of the bacteria. But consider the following. One patient might fail because they die. Another patient might fail because they simply lack microbiological eradication. Now, primary analyses, if this is binary, might treat these as equivalent. Shouldn't a primary analysis, though, recognize the important difference of these particular issues? And so can we do that? So we have moved towards a desirability of outcome ranking in some of the most common antibacterial indications. And we have standardized, thought about standardizing the door to about 5 levels. Now, in antibacterial, in these infections, what's often measured is survival, some definition of clinical response, which may have to do with symptoms and so forth, a clinical evaluation, whether the patient had infectious complications, and whether the patient had a serious adverse event. And the more of those, these are all bad things happening, and the more of those bad things that happen, the worse off the patient experience was. So we basically set up a count. How many of these things did you have? And we've got 5 levels of the door. If you survived and had none of them, then you're in the best category. If you die, you're on the bottom and you're in the worst category. But then we count up how many of these bad things happens and categorize you. Now, there's different types of infection sites. Bacterial pneumonia on the left side. Bacteremia is a blood infection. Complicated urinary tract infection on the right. Intra-abdominal infection on the far right. And the definition of a clinical response, or which sort of infectious complications we might look for, may vary by the site of infection, but there's a sort of a standardized approach now to that we're developing to develop a door in each one of these infections. What about oncology? Well, here's a motivating example. This is the Bellini trial, a very recent trial, randomized, double-blind, placebo-controlled trial. It compared venetoclax versus placebo, both of those on top of a backbone therapy. for the treatment of relapsed refractory multiple myeloma. And it turned out that this particular drug was superior to placebo on progression-free survival and a number of other things. But there was a tricky issue here in that it was actually inferior with respect to mortality. So with progression-free survival, so this brought up a very tricky issue. The drug is winning on several outcomes, but not on mortality. So one thing we could do is think about how we might set up an ordinal desirability of outcome ranking in this particular scenario, recognizing mortality as different from progression and incorporating toxicity. And so maybe it's got 4 or 5 levels, again, with the most desirable at the top, where there's progression-free survival without a toxicity, mortality's at the bottom, but there's a couple of layers in between in which some things are going right, like survival, but not everything is going right. So the new map, as I see it, if you might imagine how let's plot the number of outcomes that we have by the number of effect estimates that we have. And right now, we have lots of different outcomes in trials. We have efficacy outcomes, safety outcomes, perhaps quality of life outcomes. And then there's a number of effect estimates, which is fairly few. We generally are estimating a global effect. Now, where we would like to get to is down here, where we compose those outcomes into a global patient outcome. But then, after we've done that, try to estimate various effect, get estimates of effect based on patient characteristics and go into the personalized medicine world. So the path to get there, though, has to be correct. We're going to make progress via composition, where we characterize the disease burden for individual patients, but then progress via personalized medicine techniques to the target we want to get to, and also bring in patient preferences. So as we do this, let me show you an example about how we might do this. This is an example from the PROVIDE study, which was a prospective multi-center observational evaluation among adult hospitalized patients with MRSA bloodstream infections. And the research question was, what is the vancomycin pharmacodynamic exposure target that's associated with optimal treatment outcome? So this is a dosing problem, and which is really a very interesting and important benefit-risk problem. The more drug, well, if you get more drug in the system, then perhaps it'll be more efficacious, but it's going to be at the expense of toxicity. So how do I find the right level of dosing? So what we did is we set up a door, 5 levels here. There was a treatment success variable, and acute kidney injury is the toxicity. So the most desirable outcome is treatment success without acute kidney injury. The least desirable is death. There's a couple of layers in between where you may or may not be getting a treatment success and may or may not be getting acute kidney injury. And here are the results of this particular analysis. And what you see is the blue color on the left-hand side is treatment success with no acute kidney injury. On the far right, what you see is mortality. And the 5 bars are quintiles of dosing levels. So higher doses on the top, lower doses on the bottom. So as we evaluate what's happening in this plot, what we see is what do we gain as we move to the higher doses? Well, what we gain is not necessarily additional treatment success. We gain additional toxicity. What we see is larger intervals there of that reddish color, which represents treatment success, but with acute kidney injury, with toxicity. And so those higher doses are basically only gaining us additional toxicity. So in conclusion, I wanted to just wrap up with some ideas for protocols as we move forward. I hope that we can place increased interest on questions of a pragmatic origin to match the clinical importance and utility. One thing we might consider is if we're doing 2 phase 3 trials during development, that we devote at least one of them to very pragmatic questions. There's a benefit if we can bring benefit risk into design and conduct. I think this will greatly enhance sort of the pragmatic nature and value of our trials. Now, CIOMS actually has a working group that is going to have some of these messages about bringing benefit risk into design and conduct rather than a post hoc exercise. So along with efficacy and safety endpoints, that perhaps we should be including global assessment of patients, door-like benefit risk endpoints that represent an analysis of the patient, and build that into protocols rather than as a post hoc exercise. And that we consider pre-specified procedure for identifying patient subgroups with a positive benefit risk profile, not just based on an individual outcome. So some conclusions, think as much about the question as you do about the answer, because if we don't get the question right, the answers are going to be of far less value. Avoid being a fashion victim. The selection of methodology should be based on objectives of the study and the contextual characteristics rather than pressures to implement trending methodologies. Innovation is getting better answers. It is not a label to camouflage assumptions that potentially lower integrity. And when necessary, we might sacrifice on quantity based on feasibility constraints, but don't sacrifice on quality. So in a recent call discussing an RFA to address the quote unquote the aging question in HIV, after 45 minutes of discussion, I get the usual question that's targeted to statisticians: how many patients do we need? So I ask, well, what exactly is the aging question? Is it whether the effect of an intervention varies by age? And if that's the question, then what we're evaluating is whether age is a predictive biomarker for the intervention. Perhaps it is whether older age increases the incidence of disease. If that's the question, then we're asking whether age is a prognostic biomarker for disease outcome. Or maybe it's whether HIV-infected individuals tend to age faster relative to HIV-uninfected people. In this particular case, we're asking whether HIV is a prognostic marker for aging. So the reply I heard was yes, all of those things, a few other things too. What's the sample size? And the grant is due tomorrow. So I replied with complete confidence, 6 roles. So in quoting the well-known physicist Richard Feynman, there is no learning without having to pose a question. So I ask you whether there are any questions. I hope we can find better questions than we're finding these days. I wanted to just thank a few people who've contributed to some of these ideas. Dean Folman, Dan Rubin, Toshi Hamasaki, my friend and colleague, Chip Chambers, my friend and colleague, clinician colleague, and the ARLG and the Socrates Steering Committee. So in conclusion, I've no doubt you will enthusiastically applaud now because you're so relieved that my talk is over. Thank you very much.
Next keynote speaker is Dr. Ivan Chen. Dr. Ivan Chen has 25 years of experience in pharmaceutical industry. He is Vice President and Head of Statistical Sciences of DSS at AbbVie. In his capacity, he leads the statistics organization supporting discovery, biomarker, clinical development, and global medical affairs for all therapeutic areas. Prior to joining AbbVie, he was Executive Director of Biostatistics at Merck. Dr. Ivan Chen also has extensive experience and accomplishment in statistical communities. He's an ASA fellow. He's also a fellow of the Society for Clinical Trials. He serves as executive director of the International Society for Biopharmaceutical Statistics. Ivan is also a past president of the International Chinese Statistical Association and a past program chair and executive committee member of ASA Biopharm section. He has more than 80 publications in statistical and clinical journals. I have had this privilege of working closely with Ivan for the past 13 years. I am deeply impressed and greatly grateful to his leadership, mentorship, and inspiration. Today, we are so happy to have Ivan here at the workshop to deliver his keynote speech on statistical leadership and innovation in practice. Once again, let's welcome Dr. Ivan Chen.
Thank you very much, Yabing, for the nice introduction, but also for organizing this plenary session. I'm very happy and honored to be invited and joining Scott Evans in this session in talking about statistical leadership and innovation in practice. This is a little bit of a disclosure for my presentation, which I won't go into detail. And what I'd like to share with you a little bit is in the following outline. We're trying to give a little bit of introduction about the drug development process. And then I'm going to share a few examples of statistical innovation and leadership in action that I've seen throughout my career. And in particular, focusing on three most important contributions that statistics is doing. One is the adaptive design strategy, Bayesian paradigm, and then use of external data in regulatory decision making. And then at the end, I will give a very brief summary. So as many of you know, and I won't go into too much detail with this cartoon, a usual R&D process for a successful drug or vaccine takes multiple years, sometimes as long as 15 years, going from basic research, looking through thousands of potential chemical compounds, and then have to do preclinical testing in animals before advancing the potential compounds into clinical trial. And then we have phase 1, phase 2, and phase 3 clinical studies before we can collect the evidence and then prepare them for regulatory approval. And so that it in general takes a pretty long time, as you can see. However, many of the things that we are doing now actually help to accelerate parts of this R&D process. And as you can see, some of these innovation and leadership the statisticians brought to the bear. If you can look at it throughout this R&D process, there are many sources of data, and we call it big data, right? From chemistry labs, from genomic and proteomic modeling, the biomarkers that we do, and then when the compound go into clinical trial, there's a lot of data from clinical trial as well as safety surveillance, or post-marketing data, healthcare claims. So those are the kind of data we also call real-world data. So if you look at this, a big circle of the big data, statisticians take part in many areas and actually is playing a critical function there. Not only that, because of technology and advancement of genomic to help us to understand the disease, or use of the biomarker to guide smart R&D, and R&D process has been accelerated, especially with the COVID-19 pandemic, it really highlights some of the enormous need for innovation and accelerated development of drugs or vaccines. Just to give you an example, the COVID-19 vaccines actually was able to start a clinical trial 2 months after identification of the genome sequence. And this is a really phenomenon and a really great achievement of the whole pharmaceutical industry and the scientific community. And advancement of the vaccine program go from phase 1 to phase 3 in less than 5 months. So it really illustrates a tremendous collaboration between industry and government to shorten the development timelines, potentially to 18 months instead of years. Throughout all this, you can see the statisticians really plays an important role in R&D in different areas. For example, biomarker and genomics, design of experiments and clinical trials, statistical analysis and interpretation of data, statistical modeling and predictive analytics. And there are also more strategic planning, like product label versus clinical development plan, decision-making and probability success, and which I will go into a little bit later. Statistician also was engaging in interaction with regulatory and external experts like the FDA, NIH, CDC, or WHO, right, and key opinion leaders. And through that, there's a lot of innovation and leadership in action. And in the following few slides, I will try to go over the 3 important areas that I highlighted previously. The first one is the adaptive design strategy. And as many of you know, the adaptive design strategy as applied to clinical trial development have multiple features. It could be dose response assessment or interim stopping for utility and efficacy. It could be dropped losers or like dose selection or adjusting sample size, so on and so forth. And what a greatest leadership in actions I've seen in my career is the pharma working group that was formed back in 2005, and realizing that adaptive design at that time is just emerging, and there's really no guidance, and the technology is not well defined. But the group of the statisticians from pharma, led by Mike Cram, actually got together and we tried to socialize and form a strategic initiative and to really promote the idea of performing adaptive designs, but also engaging with other industry peers, governments, academics, to really try to explore a little bit about this adaptive design area. And the group also published a series of white papers on adaptive designs and really start to engaging the regulatory agencies, not only in the United States, but also EMA, PMDA. And you can see the statisticians down at the FDA also start to take on the leadership and develop the guidance document. And this document was dropped in 2010 and finalized in 2019. What it really talks about is that you could actually allow a variety of adaptation rules as long as adequate evaluation of operating characteristics is documented. And it really also emphasized that simulation plays an important role in understanding the trial operating characteristics using adaptive designs. One example I wanted to really highlight is this HPV vaccines. Is the phase 2-3 design with dose selection based on a biomarker. And you can see in the phase 2B area stage, there's only 4 different doses, 3 active doses and an active control. And it's relatively small in sizes to look at the immunogenicity data. And then based on the immunogenicity data, final dose was selected to go into phase 3 and then expand the trial to follow for clinical outcomes. And in the end, all those follow-up data from both phase 2B and phase 3 is included in the final analysis. And what it does is that the statistician actually took the time to conduct a comprehensive simulations to really show that with this kind of study design, the operating characteristic is well under control. For example, interval error in a wide range of scenarios. And the study design not only improved the statistical efficiency in terms of power gains, but it also reduced the white space between phase 2 and phase 3. And this is sort of one of the a really great success that it actually quoted in the FDA guidance on adaptive designs that was recently released. Another example I wanted to cite is in the area of response adaptive randomization, or RAR. And I know we know appropriate statistical method for analysis can control the overall interval error. But there remains to be a little bit of a concern about heterogeneity of the patient population from time to time. So one of the great success examples that I've seen is actually was told by Scott Berry in one of the diabetes trials that Lilly conducted. And this was in the phase 2 part of it had 7 doses plus placebo plus active control, and using this RAR, using it between 200 and 400 as a burn unit, and then to select 2 doses for phase 3. And in the phase 3 part of it, the doses use a fixed randomization, and this actually led to a very successful dose selection that was utilized for phase 3 and actually led to a very successful project for the diabetes drugs. However, the regulatory acceptance RAR in pivotal trial like phase 3 varies. I think we really need some more interaction between sponsor and regulatory agencies to have a common understanding of the risks and benefits of such designs in pivotal trials. And I think this is an area, this is a perfect for statisticians to lead the discussion. Some more recent development, obviously, around the adaptive design that we can see because of the PDUFA 21st Century Cure Act, right? The FDA actually issued a draft guidance on complex innovative trial designs. And this pilot program will enhance the sponsor interaction with the FDA on the implementation of innovative trial designs. So the broader implementation of innovative trial designs, including a master and platform trial, the Bayesian adaptive trials, and innovative use of external data. So I think continual engagement between the industry and regulatory agencies can really enrich the experience and knowledge sharing. So the second area I want to highlight is the Bayesian paradigm. As you can, we all know that the Bayesian strategy has been gaining traction because we could incorporate the prior knowledge in the design and analysis. It could also accelerate decision-making, right, with less new data by leveraging prior information. And this is really made possible because of advancement of computational power. And FDA in the device area issue a guidance on the Bayesian statistics for medical device. And recently, FDA have also discussed the use of Bayesian designs in this draft guidance on the innovative trial designs. Some of the applications I've seen in this area include in the early oncology development area, where the bond design and CRM design have been used to really adapt and leveraging the prior information that we got from the trials. So this has been shown to be having a much better performance than the traditional 3+3 design. Another strategy that I've seen being applied is in the predictive models and go, no-go decision framework. And for example, looking at a proof of concept trial or looking at a phase 2 trial in order to predict the success for phase 3. Right? There's modeling being done and using the predicted probability of success based on interim data. And we have used this techniques all the time in our sort of long-range trial planning and implementing in many of our programs at the third area I wanted to touch on is this hierarchical modeling for subgroup analysis. For example, when we look at the trial outcomes by demographic variables, and this is something the FDA have also applied this methodology to drug trial snapshot. And this is actually initiated by the FDA statisticians using this MCMC modeling to estimate the potential subgroup effects of the drug and apply the first example to this drug called Rinvoq, which is an RA drug. And you look at the web, I think FDA have highlighted this as one of the innovations statisticians bring to the table. And it's a great regulatory science impact story a couple of years ago. The third area I wanted to highlight in terms of statistical leadership and innovation is the use of external data in regulatory decision making. And if you think about the clinical trial represent really a big chunk of the R&D costs. So if we could leverage the external data in similar patient population, this could actually really significantly reduce the cost of the clinical trial. So FDA draft guidance on complex innovative trial design discuss some of these considerations that also. This slide shows you there are many different ways one could borrow the data from previous run clinical trials, right? You could maybe borrow the historical control data and add it to the current control, so sort of borrowing on top. Or you could borrow the historic control to substitute the current control. Or in another extreme, so you just completely remove the current control and just use the historical control. And all this will lead to a so-called synthetic control study. There's a very nice paper by the industry statisticians through the Translator collaborations to really look at this particular paradigm and define a really good strategy how to go about finding a suitable source for control, and how do you need to prospectively plan the selection of the external controls, and how do you actually define the optimal set of external controls. So if you are interested, please take a look at this, a very interesting paper. And I wanted to share with you one example that we went through just recently at the, this is about a proof of concept trial in immunology where we have 4 doses comparing to a placebo control. And in the design stage, we know there are some historical data from a recently complete trials. Some of those actually are from AbbVie, some of them are not from AbbVie, sort of from a drugs in the similar classes, but they are in the similar populations. So we really took a look at that and came out in selecting 3 potential studies for use. And initial plan is for about 40 subjects per group in the POC trials, but looking at this historical data, if we summarize the information using a sort of meta-analytic approach, you could see that a total of about 362 subjects could give us some information. Maybe if you're using a random effects model, the effective sample size may be about 62 or 64. So at the end, what we end up using this approach is to maybe we decided to borrow 20 subjects and then so we could reduce the concurrent control from 40 to 20. That way, you could save some of the sample size in the concurrent control, but also allow some safety comparison because you still have a randomized control in this fashion. And ultimately also save the cost and saves time for the clinical trial operations. Another particular scenario is the synthetic control. And here I wanted to emphasize that this is really referring to leveraging the patient level individual data from external source. It could be from previous clinical trial data, or it could be from real world data. And propensity score matching is a common method where you could look at all the baseline covariate and then decide as to whether some of these treatment groups are similar in characteristic. You could group them by their propensity scores. In this particular case example, David et al. Again is a group of statisticians from industry. They did a nice job illustrating that in one of the previous run oncology trial, using the propensity score matching, you could actually match really well the control group to what's in the actual trial. In the left panel, you can see before matching the historical pool and the control in these target trials, it still have some discrepancies. You look at the log-rank statistics that comparing the two groups actually have a little bit of a significant differences. But after applying this historical control data methodology to select a control, you could really eliminate that kind of discrepancies. So recently, we have actually applied this methodology in one of our RA trials in comparing a new drug to adalimumab, which is Humira. And in that trial, we actually have a new drug and then have the active control, but we don't have a placebo. So in this particular scenario, we try to use the propensity score matching to identify synthetic control from the previous completed clinical trials. And the idea is pretty simple. We use a methodology sort of based on the caliber, which is the relative difference based on all the baseline covariates and calculate their propensity score and then calculate how close they are with regards to the different neighbors. And then from there, we also look at the operating characteristics of this synthetic control matching and looking whether you could use 1-to-1 matching or 1-to-2 or 1-to-3. And actually, in this particular case, we find out a 1-to-2 actually could maximize the probability of success. And as you go on to a higher matching, it doesn't really help that much, sort of the diminishing returns in terms of power gain. So, so these are some of the examples I wanted to share with you. And in every single one of those examples, you can see that actually statistician is carrying a lot of this kind of leadership behavior and have to work closely with the study teams to really influence teams about this kind of new methodology and to really convince them that the clinical trial can be better executed with greater certainty and higher efficiency. So in summary, I wanted to really just make 2 points. Number one is that the innovative designs and analytical strategy really have increased the success rate of drug development over the last few decades. And in particular, statistical leadership in innovative thinking plays an important role. And I want to leave you all with this quote: leadership is not a position or a title, it is action and example. Okay, thank you very much for your attention.
Thank you so much, Ivan. What a wonderful talk. So exciting to see there are so many innovative practices in action. Thanks, Ivan. All right, next, let's start the Q&A for both of our keynote speakers.